Hello students. So next we shall consider the steps that enable the bacterial cells or bacteria to take up recombinant DNA. So here there are different steps that are followed, right? So that the recombinant DNA enters the bacterial cell, that is the host cell. So here bacteria, they are the host cells, right? So the host cell is the bacterium. So recombinant DNA, it has to be passed into the host cell. So to do this, there are some procedures involved. Now, why this recombinant DNA is transferred to the host cell? A very simple question, right? Why recombinant DNA or rDNA is transferred in the host cell? The main purpose is gene cloning. So gene cloning is the main purpose. Gene cloning is the main reason why recombinant DNA is transferred to the host cell. Now, these steps we shall consider, right? How it is transferred. So first, what is done? See, you consider one petri dish. So imagine this as a petri dish. So petri dish, it is a glass plate or a fiber plastic plate, right? So in this petri dish, there are E. coli. There are E. coli cells, right? Now here the E. coli cells, they are kept in a culture medium, right? E. coli cells, they are kept in a culture medium. So in the previous video we discussed culture medium. So one example of culture medium is a polysaccharide that is called agar. So in a petri dish, this polysaccharide is taken, E. coli cells are introduced in this petri dish. Now this becomes, what this becomes? It becomes a culture of E. coli cells, right? So this becomes a culture of E. coli cells, the culture containing E. coli cells. Now, the first step here is the addition of calcium. So calcium is added where it is added to the culture of E. coli cells, right? Calcium is added to the culture of E. coli cells. Now, what is the effect of calcium? Right? What is the effect of calcium on E. coli cells? Can you imagine the effect? See, a E. coli cell is having a cell wall, right? E. coli cell has a cell wall. Now, the pores which are present on this cell wall, right? Pores, very small or minute, very minute openings, right? Very minute or very small openings, they are called pores. So pores are present on the cell wall of E. coli cells. This calcium, it has direct effect on the pores. So due to the effect of calcium, what happens to the pores? Pores become wide. So there is widening of pores. There is widening of pores. So pores which are very narrow, now they become wide, right? So what is the use of this effect? So when pores become wide or when pores become large, the recombinant DNA, it can easily enter the bacterial cell, right? So that is the main purpose of using calcium. It widens the pores on the cell wall of bacterial cells. This helps recombinant DNA to enter inside the E. coli cells, right? So after this, the next step, recombinant DNA is added to the culture of E. coli cells, right? The recombinant DNA is added to the culture of E. coli cells. Next, what is done? The temperature is reduced, right? The temperature is reduced. How? So incubation on ice is done, right? This culture medium is incubated on ice, right? It is kept on ice to reduce the temperature. So here temperature is extremely reduced, right? So this extreme low temperature, it favors the entry of recombinant DNA into the host cell. Next step, after incubating on ice, the next step is to give a shock that is called heat shock, right? After reducing the temperature, next heat shock is given. That means the temperature now is raised to 42 degrees Celsius. So the culture of E. coli cells, now it is suddenly exposed, right? The culture of E. coli cells, now it is suddenly exposed to higher temperature of 42 degrees Celsius. Right? Initially the temperature was low when it was incubated on ice. Now suddenly they are exposed to high temperature of 42 degrees Celsius, and this step is called heat shock. So heat shock is given for a brief time or for some time. After heat shock, again the culture of E. coli cells is incubated on ice. After heat shock, again the culture is incubated on ice. So all this treatment, right? All this treatment, initially addition of calcium, then reducing temperature by incubating on ice, heat shock, again reducing the temperature by incubating on ice. So all this treatment, what it does, it causes the entry of recombinant DNA into the bacterial cells. So this is how recombinant DNA enters into bacterial cells, right? And later on what happens? Bacterial cells multiply. One bacterial cell produces 2, 2 produces 4, 4 produces 8 after every 20 minutes. And the recombinant DNA which is present now, recombinant DNA which is present in the bacterial cells also undergoes replication, right? So this recombinant DNA replicates. Along with that, gene of interest or desired gene also replicates and gene cloning takes place, right? So these are the various steps which are used to enable bacteria to take up recombinant DNA. So this is how recombinant DNA is made to enter E. coli cells. So you may expect a question, maybe for 2 marks or a 3-mark question. Mention the steps to enable bacteria to take up recombinant DNA. So these are the steps. First is calcium, then incubation on ice, heat shock, again incubation on ice. So by these treatments, recombinant DNA enters the host cell and gene cloning takes place, right? So I hope these steps are very much clear to you. Then next we will consider the methods to introduce alien DNA into the host cell. So there are different methods, right? There are different methods to introduce alien DNA into the host cells. Now the question is, what is this alien DNA? So alien DNA is the foreign DNA or the gene of interest or desired gene or the selected gene. So to introduce this desired gene into the host cells, they may be plant cells, they may be animal cells, they may be bacterial cells, right? So all these cells where foreign gene is transferred, those cells are called host cells. Understood the meaning of host cells? The cells where desired gene is transferred, they are called host cells. So the methods we shall consider one by one. So the first method is the micro injection. The next biologics, right? The second method is biologics. The next one the third is the disarmed pathogen vectors. So these are the 3 important methods to introduce alien DNA into the host cells: microinjection, biologics, disarmed pathogen vectors. So you may expect a 3-mark question. Mention the methods to introduce alien DNA into the host cells. So just you have to mention these 3 methods. So first we will consider micro injection. So what is this micro injection? It is a technique to transfer the alien DNA or the recombinant DNA, right? It is a method to transfer recombinant DNA into the animal cells. So recombinant DNA is directly micro injected into the nucleus of animal cell, right? See again, micro syringe is taken. Micro syringe is taken. With the help of this micro syringe is something that is used to inject medicines, right? or in simple words you call it as an injection. So a micro syringe is taken. With the help of micro syringe, recombinant DNA is directly injected into the nucleus of animal cells. So this is how the desired gene through recombinant DNA is transferred into the nucleus of animal cells. This method is called micro injection. Desired gene is or recombinant DNA is directly micro injected into the nucleus of animal cells. Then biolistics. See, biolistics is also called the gene gun. Right? Biolistics is also called gene gun. So biolistics or gene gun is a technique to introduce alien DNA in plant cells. Method to introduce alien DNA in plant cells. It is called biolistics or gene gun. So what is done here? See here the microparticles, right? The microparticles of gold, right? Microparticles of gold or even tungsten microparticles of gold or tungsten, they are coated with DNA, right? They are coated with DNA, and these microparticles, they are bombarded on plant cells, right? So they are bombarded on plant cells with a very high velocity, right? Microparticles of gold or tungsten, they are coated with DNA and they are bombarded on plant cells with a very high velocity. So what happens with this? These microparticles, they enter the plant cells, they enter the nucleus of plant cells. Along with them, they carry this DNA, right? So this is how we can transfer this DNA into the plant cells, right? So I'll try to make it little simple. These microparticles of gold or tungsten, they are coated with DNA and they are sprayed on plant cells, right? They are sprayed on plant cells with very high speed or with very high velocity. Just like a sprayer. Here, you take water in a sprayer and you spray it on the plants. So that water comes in contact with plants at a very high speed when you spray it with a sprayer, right? Same thing is done here. Microparticles of gold or tungsten, they are coated with DNA and they are sprayed on plant cells with a very high speed. So these particles, they enter the nucleus, along with them they carry the DNA. And in this way, DNA is transferred to the target site. This method or technique, it is called biolistics or a gene gun, right? How we shoot a bullet, right? We shoot a bullet by using a gun. So here, these microparticles are like bullets, right? Which are shot on plant cells. And these bullets, they reach the target site. Means these particles, they reach the target site, that is plant cells. Along with them, they carry the DNA. Fine. The next, disarmed pathogen vectors. Disarmed, already you know the meaning. Disarmed means to make a particular organism harmless, right? It is called disarmed. Pathogen. Pathogen is an organism that causes diseases. Any organism that causes a disease, it is called pathogen. So disarmed pathogens means these pathogens are made harmless. A harmless pathogen is called disarmed pathogen. So this pathogen now it cannot cause a disease because it is modified. So a modified pathogen will not cause disease. So such modified pathogens or such modified microorganisms, they are called disarmed pathogens. Now these pathogens or microorganisms like the virus and bacteria, right? Now they are used as vectors. So these virus or bacteria, they are disarmed, they are made harmless, and now they are used as vectors to transfer alien DNA into the host cell, right? So these are the methods, right? These are the methods to transfer or to introduce alien DNA into the host cells. So very important part. So you may expect a 1 mark question, what is micro injection? Or you may expect a 1 mark question, what is gene gun? Right? Or you may expect a 3 mark question, mention the methods to introduce alien DNA into the host cells. So I hope you have understood this concept. So we shall stop and I will continue in the next video. Thank you.